Hey there! Today I will be solving problems that can exhibit my understanding of a non-Mendelian cross. I will highlight the information I obtained this week pertaining to incomplete dominance, codominance, and multiple alleles. I am additionally going to do my best to thoroughly explain a combined dihybrid slash non-Mendelian cross problem. Let's commence with the first question. The first problem is an example of incomplete dominance. So incomplete dominance transpires when two heterozygous alleles are manifested together in the phenotype of an organism. The distinct phenotypes generated by each allele are blended together into a third phenotype. The dominant allele essentially is unable to conceal the effects of the recessive allele, and the organism's resulting physical appearance is a blend of both alleles. Essentially, incomplete dominance is when neither gene is completely dominant and the outcome is a brand new trait. The question itself is, if the red color in Zynas is incompletely dominant over white, how many pink flowers will be in the F1 generation if a red Zyna and a white Zyna are crossed? What are the phenotype and genotype ratios of the F2 generation if two pink flowers are crossed? To begin solving this problem, there is a series of steps I must follow. First off, I'm going to define the variables, which are the letters that will represent the alleles. I will use a capital Z to represent the red color in the Zynas as dominant alleles are capitalized, and I will use a lowercase z to represent the white color in the Zynas as recessive alleles are in lowercase. A homozygous red Zyna will have two capital Z alleles as they are the same, and a homozygous white allele will have two lowercase z alleles. The outcome of these two Zynas being crossed can be seen in the F1, so the first filial generation on the Punnett square. I've gone ahead and already created the Punnett square, and as you can see, the results distinctly show that all flowers will be capital Z and lowercase z and pink. So if they're crossed, that's the outcome that's going to be seen. This again ties in with incomplete dominance, as the red Zynas are incompletely dominant over white, which therefore leads the offspring to be heterozygous, capital Z, lowercase z, with pink flowers. And this was only the first portion of the question, and now we're going to move on with the second part. So the second portion of the question involves determining the phenotype and genotype ratios of the F2, so the second filial generation, if two pink flowers were crossed. So I followed the exact same steps and created a Punnett square in order to determine the answer. I placed two heterozygous, so capital Z and lowercase z, pink flowers on both sides of the Punnett square, and this resulted in one pair of capital Z, two pairs of capital Z, lowercase z, and then one pair of lowercase z. The phenotype ratio of the F2 generation when two pink flowers are crossed is 1 to 2 to 1 of red to pink to white. And the genotype ratio would also be 1 to 2 to 1 of capital Z, lowercase z, and lowercase z. The percentages are also listed below of 25% of the flowers could be red, 50% of the flowers could be pink, and 25% of the flowers could be white. Stay tuned as I'm going to create a formal conclusion for this question. In conclusion, if the red color in the Zynas is incompletely dominant over the white, and if they are crossed, that would result in all pink flowers, so capital Z, lowercase z. The distinct phenotypes generated by each allele are blended together into a third phenotype, which is the pink color of the Zynas. And then moving on to the second part of the question, and that relates to the phenotype and genotype ratios of the F2 generation if two pink flowers are crossed, which would be 1 to 2 to 1 of red to pink to white, which is the phenotype, and capital Z to capital Z, lowercase z, which is the genotype. So the next problem is an example of codominance. It remains imperative to recognize that incomplete dominance is not the same as codominance. In codominance, both alleles can be seen in the phenotype at the same time. For instance, instead of being uniformly pink, a flower with red and white alleles that show codominance will have both patches of red and white. Simply put, the distinct phenotypes produced by each allele can both be observed. Neither allele is exactly dominant, and the result is an entirely different phenotype. The prefix co means together, and many individuals use that to identify that both alleles show up together. The question itself is, in some chickens, the gene for feather color is controlled by codominance. The allele for black is B and the allele for white is W. The heterozygous phenotype is known as an erminet, black and white spotted. A. What is the genotype for black chickens? B. What is the genotype for white chickens? And C. What is the genotype for erminite chickens? The second part of the question is as follows. 2 erminite chickens were crossed. Show the Punnett square. A. What is the probability that they would have a black chick? And B. What is the probability that they would have a white chick? So the first part of the question was pretty easy, as essentially it's just defining variables. So A asks for the genotype for black chickens, which I defined as BB, as different variables are used in codominance problems, so I couldn't use the same one for black and white chickens. B asks for the genotype for white chickens, which I defined as WW, so that the distinction between them is clear. C asks for the genotype of the erminate chickens, which I defined as BW, as the chickens will be speckled as both alleles are simultaneously being expressed in the heterozygote. So to begin solving the next part of the problem, there were a series of steps I needed to follow. I started by placing the 2 heterozygote BW erminate chickens on both sides of the Punnett square, and this resulted in 1 pair of BB, 2 pairs of BW, and 1 pair of WW. The phenotype ratio of this generation is 1 to 2 to 1 of black chick, black and white speckled chick, and white chick. The genotype ratio would also be 1 to 2 to 1 of BB, BW, and WW. Based on these numbers, I can calculate the probability of them having a black and white chick. The probability that they would have a black chick is 25%, which is the same as the probability of them having a white chick. There is a 50% chance that they would produce a chick with specks of black and white based on this information gathered in the Punnett square. In conclusion, the probability that 2 erminate chickens would produce a black chick is 25%, and the probability that they would produce a white chick is also 25%. There is a 50% chance that they would produce an erminate chicken, as you can see in the diagrams below. So this next problem is an example of multiple alleles. Multiple alleles is a type of non-Mendelian inheritance pattern that includes more than the typical 2 alleles that usually code for particular characteristics in a species. In multiple alleles, there are more than 2 phenotypes possible depending on the dominant or recessive alleles that are available. The human ABO blood types are a good example of multiple alleles, which brings me to introduce the next question. The question is as follows. A wealthy elderly couple dies together in an accident. Soon, a man shows up to claim their fortune, contending that he is their only son who ran away from home when a boy. Other relatives dispute this claim. Hospital records show that the deceased couple's blood types were AB and O respectively. The claimant to the fortune was a type O. Do you think that the claimant was an imposter? Explain. So in order to tackle this question, I must find out whether it is genetically possible for the claimant man to be the son of the elderly couple. I can do this with the help of a Punnett square. To begin, I must define the variables with respect to the phenotype and genotype. The phenotype, which is an appearance of an organism based on the genotype of AB blood, is AB. The genotype, which is the genetic makeup of an organism, for type AB blood is IA-IB. The phenotype for type O blood is O, and the genotype for type O blood is II. Next, I write the parents' genotypes on the top and side of the Punnett square, and then I continue to fill it in. As you can see, this results in only 2 possibilities one being IAI, which leads to be blood type A, and the second being IBI, which leads to be blood type B. In conclusion, the claimant is an imposter, and there is no way he could have been the genetic son of the elderly couple. This is because one parent contributes the I gene and the other contributes the IA or IB to any offspring. When combined, this can only result in blood types A and B, therefore making it impossible for their child to have blood type O. Since the imposter has blood type O, it unquestionably concludes that he could not be the child of the deceased couple. We have now reached the last problem I will be doing, which is an example of a combined dihybrid / non-Mendelian cross. A dihybrid cross is a cross between two different lines / genes that vary in two observed traits. To solve the following question, I reference additional sources such as the Amoeba Sisters YouTube video on dihybrid and two-trait crosses with the use of the FOIL method to help with my problem-solving method. The question is as follows. Suppose that in a species of flower petal color can be red, white, or pink, and petal texture can be wrinkled or smooth, with wrinkled being the recessive trait. What is the likelihood of a pink wrinkled flower resulting from a cross of a red wrinkled parent and a pink homozygous smooth parent? You must combine all the data into a single Punnett square. To begin solving this problem, there is a series of steps I must follow. First off, I'm going to define the variables, which are the letters that will represent the alleles. The dominant allele for a smooth flower petal will be represented by capital T, lowercase t. The recessive allele for a wrinkled flower petal will be represented by lowercase t, lowercase t. The dominant allele for a red flower petal will be represented by capital C, lowercase c, and the recessive allele for a pink flower petal will be represented by lowercase c, lowercase c. Mendel's law of segregation states that each gamete is going to have two alleles. In those gametes, you have to account for every possible combination. Mendel's law of independent assortment says that those alleles are not linked. This is where the FOIL method comes into play in determining the gamete combinations. FOIL stands for first, outside, inside, and last. So, capital T, lowercase t, capital C, lowercase c, and lowercase t, lowercase t, lowercase c will be crossed, and the combinations will be as follows and as follows. So it would be capital T, capital C, capital T, lowercase t, capital C, and lowercase t, lowercase c, and all these are lowercase. And then next up, I'm going to be plugging this into a Punnett square. I've gone ahead and plugged the combinations into the Punnett square to determine what the offspring prediction will be. As you can see, the genotype and phenotype ratio is the same 1 to 1 to 1 to 1. To further elaborate, the genotype of capital T, lowercase t, capital C, lowercase c is 25%, it's 4 out of 16. Capital T, lowercase c, lowercase c is also 25%, 4 out of 16, which is the same with the other two combinations. What about phenotypes? Well, 25%, so 4 out of 16 of the flower petals will be smooth and red, 25%, so 4 out of 16 will be smooth and pink, 25%, so 4 out of 16 will be wrinkled and red, and lastly 25%, so 4 out of 16 will be wrinkled and pink, which was what the question wanted to know. In conclusion, the probability of a pink wrinkled flower resulting from a cross of a red wrinkled parent and a pink homozygous smooth parent is 25%, so 4 out of 16, which is an equal chance as any other combination. The end. Thank you for listening. I hope I was able to guide you through my problem-solving strategy while displaying all the information I obtained this past week. Hope you had fun.